I'm going to show you two animations of transcription happening because I think the more visuals you see of this process, the more it's going to make sense. So in this video, the purple is the RNA polymerase enzyme. The blue double helix here is the DNA. One thing to notice here is that transcription doesn't start at the very end of a DNA molecule because one long DNA molecule could contain hundreds or even thousands of genes. And so this little region that's highlighted here is the start of the gene, okay? So when the cell wants to make a particular protein, a signal will tell the RNA polymerase to bind at that spot. So that's what we're going to see happen. So now the RNA polymerase is attached to the DNA. And as the RNA polymerase moves along, it's going to transcribe the gene. So you can see all of the RNA nucleotides floating around there. They are just in your nucleus. And so as the RNA polymerase moves along, it unzips the DNA. And now you can see the RNA nucleotides coming in into the RNA polymerase enzyme and pairing up with the DNA. So on the bottom here is the DNA. Notice the blue. So in this video, they're representing the deoxyribose backbone with blue, and then the ribose sugar backbone with red. Okay, so the blue backbone is the DNA, the red backbone is the RNA. And so notice if the DNA sequence was TGA, then RNA A's will pair with T's, RNA C's will pair with G's, and RNA U's will pair with A's because RNA does not have T. So just notice that as this plays along, you're always seeing orange match with yellow and purple match with blue. And eventually the RNA polymerase will reach the end of the gene, and then that RNA molecule will detach. So I want to show you another video because the video you just watched was essentially in slow motion. This video is intended to be a more realistic computer animation of what's happening. So here is the RNA polymerase already attached at the start of the gene. There's a bunch of proteins that kind of help the RNA polymerase assemble itself and get started. And then a little start signal is going to come down and tell the RNA polymerase to start moving. And this is how fast it actually happens in real life. So the blue is the RNA polymerase moving down the DNA. The pink is the DNA. You can see all the RNA nucleotides. Those are the little yellow pieces. And then notice how fast that RNA molecule is being built. When they zoom in again on the RNA polymerase, you should be able to see how the DNA is like unzipping and rezipping. And there's like a little hole where the RNA nucleotides go in. So isn't this insane how fast this happens? It is so cool. And then once the RNA polymerase gets to the end of the gene, you'll have this really long piece of RNA that will be ready to leave the nucleus because that RNA is carrying a copy of the message. And then the ribosome will use that RNA to make the ribosome will use the RNA to make proteins. So I think it ends pretty soon. Looks like we're almost to the end. There you go. Okay, so that also gives you a sense of how long a gene is. In the little foam model that I have, it's like 40 base pairs long. A real gene would be hundreds or thousands of base pairs long. So that's why we watched it for so long, even though it's like happening really, really fast. It takes a couple of minutes to copy a single gene.